Hi guys, so Jason Matthew here, and today we're going to be looking at Neuroscience 2, and we're going to be dealing with the action potential. So throughout this lecture, we're going to be having some crude and sexual humor, and we're going to talk a little bit about some drugs. So it's PG-13 people, so just in case. Right, so what we did in last class? Well, in the last class, we looked at the structure of the neuron. Today, we're going to be doing a few things. We're going to be looking at the resting potential. We'll see how an action potential is made. Look at the refractory period. And we're going to be talking about the transmission of action potentials, as well as the speed of an action potential. How fast does it go, and how we could change that. All right, so the lipid bilayer of the membrane is impermeable to ions, and you learned that in your very first class in cell biology at year 1. Ions can only move through the membrane via channels and pumps. And when we are talking about neurochemistry and the creation of an action potential, there are two channels and one pump that we need to look at. We have the sodium-potassium pump, which is very important. And we have the voltage-gated sodium channel, and we have the voltage-gated potassium ion channel. All right, so these are the three guys that are responsible for the different stages of the resting potential, and they're going to the action potential. You're going to be seeing how these three guys, how they play a role in that. So what we're looking at here is the sodium-potassium pump. And there's some very important stuff happening here. Alright, with the sodium-potassium pump, you are pumping 3 Na plus to the outside of the cell. At the same time, you're bringing in 2 K plus or 2 potassium ions into the cell. And what we say here is that the sodium-potassium pump, by doing that, by transporting 3 Na plus out of the cell and 2 K plus into the cell, it establishes a negative membrane potential, and we say that difference in charge distribution here makes this sodium-potassium pump electrogenic. Now, the typical resting membrane potential will be around minus 70 millivolts. Now, we call this the sodium-potassium pump, and the word pump should tell you that you have to use ATP. All right, you're going to use ATP. One molecule of ATP will be converted to ADP plus PI, and the energy given off will be used to pump 3 Na plus out and 2 K plus in. So what that is telling you also is that we need energy because we are moving these ions against their concentration gradient. If you're moving ions against their concentration gradient, then that transport will be active transport. Alright, so this sodium-potassium pump is responsible for the resting potential of around minus 70 millivolts. Now, what I have done for this lecture is that I have blocked out certain key words. And what I want you all to do then, in this slide as well as in subsequent slides, is to try to pause the YouTube presentation here and go through the whole thing and try to figure out what could possibly be those words missing there. So that will enhance your learning experience. So let's go. So we're saying ion channels. All biological membranes, including the neuronal membrane, are composed of a lipid bilayer that has an electrical resistance. That is, ions will not really flow through it. So the key thing is, what do you think? It's a choice between, is it high electrical resistance or is it low electrical resistance? So if you said high electrical resistance, you'll be correct. An ion channel is a protein macromolecule that spans a biological membrane. And we say that we call it a certain type of pore. Alright, and the key thing here is that we're saying it's a protein that is spanning the whole membrane. So So what do we know from our protein chemistry and so on, from our membranes? What is a protein that spans the whole membrane? What do we call that? Exactly, we call that a transmembrane. Alright, so let's read it over. An ion channel is a protein macromolecule that spans a biological membrane, so we call it a transmembrane pore, and allows ions to pass from one side of the membrane to the other. The electrical gradient determines the direction in which the ions move. Alright, so there are two types of ion channels. There will be the voltage gated. An opening is achieved by changing the voltage across the membrane, for example, on the arrival of a, so what do you think it will be? Right, it will be an arrival of an action potential. And then there are the ligand gated ion channels, which open when a chemical, alright, so in the second one is a chemical, not a voltage. So a ligand gated channel will be one that opens when a chemical such as, and if you said neurotransmitter, you'd be correct, such as a neurotransmitter binding to it. Now this is a pretty hardcore division here. We have voltage gated and ligand gated. And in nature, sometimes, there's that gray area. So what we're saying below there is that not all ion channels can be easily classified into these two categories. Because there are some voltage gated channels that can be modulated by neurotransmitters as well as by calcium ions, while some ion channels are not opened by change of voltage or by a ligand, but instead are opened by mechanical stretch or pressure, for example, the somatosensory and auditory receptors. The flow of ions through the channel can be either selective or non-selective. If the channel is selective, then it only allows certain ions through, and it achieves this by means of a filter. The selectivity filter is based on thermodynamic concepts and gives the channel its name, example, sodium channel. And we're going to be spending a lot of time in this lecture on the structure of a sodium channel as well as the way in which it works, the mechanism behind it. So we're going to be spending a lot of time on the sodium ion channel. However, certain channels are non-selective in that they allow many different types of similarly charged ions through. Alright, example, the acetylcholine cation channel. So from now on, if you see ACH, that's the symbol for, or the abbreviation for acetylcholine. So far, let's summarize some of the stuff that we have learned. What are the fundamental properties of an ion channel? Well, one, it must be a transmembrane pore, because basically what you're doing is going to be taking ions either from the inside of the cell and putting them outside, or you're going to be taking ions from outside of the cell and transporting it inside. So the only way that could happen is if this protein, this ion channel, spans the whole membrane. So it's a transmembrane pore. Alright? They are open. They go from an open to a closed state, and from a closed state to an open state, right? So they go back and forth from an open to a closed and a closed to an open. Because sometimes you want these ion channels open, sometimes you want them in a closed state. So it has to be going back and forth. Right? And how when they open and they close will depend on the response to a specific stimuli. So you have a sensor for voltage change. Alright, so this voltage sensor will be for like a voltage gated ion channel. While if you are a ligand gated ion channel, well then you'll have receptors on the ion channel that ligands, particular specific ligands, will bind to. Alright, so let's spend some time here. This is a biochemistry course after all, so we need to know the details of the structure of the ion channel. Now, the ion channel is made up of a few subunits, but the one that we want to pay particular attention to in terms of for neurochemistry will be the alpha subunit. Now, the alpha subunit is made up of 4 domains. Now, if you really look at this picture, there's all the information there in terms of the description of the structure of a voltage gated ion channel. The voltage gated ion channel, the sodium ion channel, is made up of 4 domains. So you have 1, 2, 3, 4. So they're made up of 4 homologous domains. Alright? Now if you look carefully at each one of these domains, each domain is made up of 6 helices. So you see they count them here for you. So you have 1, 2, 3, 4, 5, and 6. So let's recap so far. The sodium ion channel is made up of, the alpha subunit is made up of 4 domains, 4 homologous domains. And each domain is made up of 6 alpha helices. So you have 1, 2, 3, 4, 5, and 6. Now let's go ahead and give some more description here. So you see they have color-coded certain helices, and there's a reason for that. So like for instance, this one, which is in blue, what number helix would that be? Just count them. It's 1, 2, 3, 4. So this blue one is helix 4. Now if you see helix 4, helix 4, helix 4, helix 4, what we are saying is that if helix 4 on each domain, right, makes up, and you see here, the voltage sensor. So each one that is in blue, which is helix 4 in each domain, makes up the voltage sensor. Alright, and if you look at red now, let me ask you, what you would describe this red component to be? Well, you could call it the segment that is connecting helix 5 and 6. So the segment that is connecting helix 5 and helix 6 in all 4 domains will come together to form the selectivity filter. Alright, and then in orange now, which will be what? Yes, that'll be helix 6. So helix 6 in all 4 domains will make up the activation gate. Now so far, you have a component in each domain. But for the inactivation gate, and this is where you have to be very careful in the exam as well, is that the inactivation gate is that segment that connects domain 3 and domain 4 together. So the inactivation gate is only this one here, alright? Which this segment that connects domains 3 with domains 4. So be very careful with that. That is the inactivation gate. Alright, so that is more or less the summary there of the structure of a sodium ion channel. So this slide here now is to see if you could remember what we just talked about. If you think you're not confident enough to describe an ion channel on your own, please go back to the previous slide and look at it again, and then come to this. This is a slide now, a test to see if you could remember what you have done so far. So please, if you don't think you're ready, to go back and revise it, and then come to this, because this is a true test. Don't just go through the motion. Go back, revise it, and then come back here. So let's see if you're ready. The sodium channels of different tissues and organisms have a variety of subunits, but only the principal subunit. And what will be the principal subunit of the ion channel? That's right, it's the alpha subunit. The alpha subunit is a large protein with some number of homologous domains. How many domains is the alpha subunit made up of? Alright, think about it. I know you know the answer. Correct. 4. You're going good. So let's continue. How many helices, how many transmembrane helices makes up one domain? Alright, I know you know the number. And that's right, 6. So that's 6 transmembrane helices in each domain. Alright, so I'm hoping that so far everybody has gotten everything correct. If some of you unfortunately didn't get something right, go back again to the previous slide and revise it again, and then continue. So let's go. Helix 4 in each domain, so that'll be the one that was blue in the diagram in the previous slide, is so what we call helix 4. What does helix 4 in each domain make up to give? That's correct. It's the voltage sensor. Helix 6, which was orange in the diagram, is thought to be the what? That's right, the activation gate. The segments between helices 5 and 6, which is called the pore region, form the selectivity filter. And the segment connecting domain 3 and 4 is the That's right, the inactivation gate. So again, if you got some of these things wrong, that's fine, you're not learning it. Please go back to the previous slide, go through it again, then come back here and try it once. And I'm sure by the second or third try, you'll be good to go. Right, so in the previous diagram when we looked at the sodium ion channel, it was all spread out, it was flattened out. Now if we look at it from a three-dimensional view, you see how all these subunits and domains and so on are interacting with each other to form this three-dimensional pore now. Alright, so the four domains are wrapped about a central transmembrane channel lined with, so in other words, it's either a choice between polar or non-polar amino acids. So if you have Na plus ions passing through this pore, what kind of amino acid do you think will line the channel? Alright, so I'm hoping that common sense prevailed here and you said polar. Alright. The four pore regions, which is red in this diagram, come together near the extracellular surface to form the selectivity filter, which is conserved in all sodium ion channels. The filter gives the channel its ability to discriminate between Na plus and other ions of similar size. So like for instance, like this ion channel here, this sodium ion channel will only allow Na plus to pass through. Another ion that is very similar in size and charge, like for instance, potassium ions. Potassium ions will not be allowed to pass through because of the selectivity filter. Alright, and then lastly, the inactivation gate, which is this guy here, alright, closes soon after the activation gate opens. So between that time between the activation gate opening and the inactivation gate closing is a few milliseconds you're talking about here. So it goes from this here to this region here. So you see if you watch it, it blocks the pore. So the inactivation gate will close it. Alright, so let's go ahead now. Alright, so we're looking at how the mechanism now behind how this ion pore opens. Alright, so remember we have let's go ahead. The voltage sensing mechanism involves the movement of which helix? Alright, I know you know it, so give me the number. That's correct, it's helix 4, which is blue. So this is helix 4 here. So these two here, alright, helix 4 is what we call the voltage sensing mechanism. Alright, and you can see what charge is it. So the voltage sensor is positive, and this is helix 4. Alright, it's perpendicular to the plane of the membrane in response to a change in transmembrane potential. As shown at the top, the strong, and I already said it, what charge is the helix? Right, so you're correct, it's the positive charge on the helix 4, allow it to be pulled inward in response to the negative membrane potential on the inside. So remember, at rest, in a resting potential, the outside of the membrane is positive while the inside of the membrane is negative. And can you remember what is maintaining this membrane potential at rest? You're correct, it is the sodium potassium pump. So when this membrane is in its resting potential, these voltage sensor, these positive helices, helix 4, alright, is pulled inward, is attracted to this negative charge here. Alright, so that's at rest. But then there will be depolarization, and depolarization is where the inside now becomes less negative, it becomes positive here. Alright, so when that happens, when depolarization takes place, helix 4 is no longer pulled into the inside here. It relaxes, it moves outward. Right? And by moving outward, this movement is communicated to the activation gate, which is these guys here. So by the voltage sensor moving out, or pulling up, alright, moving outward, alright, the activation gate undergoes a conformational change, meaning it changes in shape, and that opens the channel, alright? And all of this takes place in response to depolarization. And in case you don't know, depolarization is when the membrane potential goes from being slightly negative, which is minus 70 millivolts, it goes to positive. All right. So let's look at it here. All right. So we have the two ion channels. We have a voltage gated ion channel, and we have a chemically activated ion channel, or a ligand gated ion channel. All right, and we won't talk too much about this one right now. We're going to talk about it a little later on, but for now, we won't talk about the ligand-gated ion channel. Just know that like for instance, this one is a acetylcholine ion channel, and you see that there's two molecules of acetylcholine will bind to the receptor here, and the ion channel will open. But we're not going to be talking about this. In fact, we won't talk about this in this lecture. We'll talk about this in Neuroscience 3 when we look at the synapse. For our purposes for this lecture, we're going to be focusing on the sodium ion channel, because this one is the one that is going to be responsible for the production of an action potential. So we're going to really focus on this one today, and in the next lecture we'll look at the ligand gated ion channel. So again, so we have right now the membrane is at rest, and you see that the voltage sensor here is blocking any ions from passing through. So no ions could pass through here because of the voltage sensor. It has the activation gate here closed. All right, and we could call this space here where the selectivity filter will be. All right, so here again is your sensor. This in red here will be your activation gate, and this one here will be your inactivation gate. So once the activation gate is across like this, there's no way any sodium ions could pass through. So this will be in the resting state. So in the resting state, that's when the ion channel is closed. No sodium ions could flow through because the activation gate is across here. All right. Now when the sodium ion channel is activated, in other words, the activated state means open. All right. The voltage sensor is going to first sense this change in depolarized. We've seen that the membrane is being depolarized, and when the voltage sensor senses that, the activation gate opens. And when the activation gate opens, Na plus ions come through. Now what I should make clear from now is that when there is depolarization taking place, both the activation gate as well as the inactivation gate is turned on. So they both start to move. But here's the thing. The inactivation gate moves a lot slower than the activation gate. So the activation gate immediately opens, but the inactivation gate has a slower kinetic, so it moves much slower than this activation gate. All right, so that's the difference there. So again, they are both turned on by depolarization. So they're both turned on the same time. The difference here is that they differ in kinetics. So this one, the activation gate moves faster than the inactivation gate. So sodium ions passes through. Now after a few milliseconds, you see what happens here. The inactivation gate comes in and blocks the pore. So no more Na plus ions could pass through. We say then that the ion channel has been inactivated. So let's see if you remember all of that. So activation of the voltage sensor or chemical receptor leads to the opening of a gate within the channel, which allows ions to flow through the channel. Inactivation involves a second gate moving into the channel. And what do you think is that word that is missing there? So read the rest of the sentence and see if you can figure out what word is that. So let's go. Inactivation involves a second gate moving into the channel more slowly than the activation gate moves out, so that there's a time when there is no gate in the channel and ions can flow through it. Alright? But finally, when the inactivation gate comes in there, it's going to block the flow of ions. It's just that it takes a lot. I know it has a slower kinetics than the activation gate, but it finally gets there. But when I say more slowly, I mean it's milliseconds we're talking about here. Right, so I talk about the sodium ion channels, but there are also potassium ion channels. And basically, it's almost the same mechanism, you know. It's just that the sodium ion channel, they also respond to depolarization, but they respond a little slower than the sodium ion channels. And you'll see very soon that that difference in response will lead to something called the refractory period and hyperpolarization, which we come into in a little while. Alright, so the sodium and potassium ion channels, those are the two main players in the next set of slides that we're coming up to. Right, so before we go there, let's learn some definitions. Alright, so when we are talking about the neuron, current is defined as the net flow of ions through a channel. Oh, that's the next thing, eh? You know, we talk about electrical impulses and so on, but please do not get confused. It's not like the electricity wires where you have a flow of electrons. This is not how the current is flowing. The current is not flowing as a flow of electrons. The current is being produced by the net flow of ions. So current is defined here for neurochemistry in a neuron as the net flow of ions through a channel. Conductance is the ease with which the ions can pass through the membrane, and permeability is the rate of transport of a substance or ion through the membrane for a given concentration difference. So what I want from you all is that you all learn these three definitions, right? So these are definitions that you need to know. So please, before you go ahead, learn these definitions. And you know, it's quite simple, you know. Look at them, they're quite easy to know. Right, so this is a little tester. Tell me what current is. Alright, so write it down. So you should have your pen and your paper there. Write down what you think is the definition for current. Right, so you should have correctly said net flow of ions through a channel. So you have one correct so far. Let's go to the second one, conductance. Again, you have that paper and pen next to you. Write down what you think what is the meaning of conductance. Very good. So you got the second one correct. The ease with which the ions can pass through the membrane. So you have 2 out of 3 correct so far. So we're going for the third one, going for the hat trick. Alright, and I know they're ready for this thing. Permeability. It is the rate of transport of a substance or ion through the membrane for a given concentration difference. Right, so we have done, you know, a good bit of work so far. So let's pause for a cause and just chill for a little while. You could go through some interesting quotes about the brain. Alright, so have a little fun there. Alright, and I don't know about you, but I always find this one to be the funniest, right? How is education supposed to make me feel smarter? Besides, every time I learn something new, it pushes some old stuff out of my brain. Remember when I took that home winemaking course and I forgot how to drive? And it's from the great Homer Simpson, right? So, please, enjoy. Right, so now we go to some real hardcore stuff here. We're going to look at how our action potential is produced. And many an exam question has been set on this very same thing that is in front of us. Alright, so let's see if we can break it down. Now, the different stages here, if you look carefully, right? There's where the neuron is at rest, and then this will represent the action potential, and here will represent basically the recovery phase. So we're going resting potential, action potential, recovery, and there are a few things going on here. Now, the first thing is across the membrane of any neuron, on the outside, so that's what extracellular means. On the outside of the cell, you're going to have a high concentration of sodium ions, and you're going to have a low concentration of potassium ions. That is extracellular on the outside of the neuron. On the inside of the neuron, intracellular, you're going to have a high concentration of potassium ions, and you're going to have a low concentration of sodium ions. So note the difference. Now, when the neuron is at rest, in other words, the resting membrane potential, when it's at rest, you have the sodium-potassium pump. This guy here is mainly responsible for the resting membrane potential. And if you remember from the first set of slides, we're saying that the sodium-potassium pump pumps 3 Na plus to the outside, and it brings in 2K plus. Alright? And the resting membrane potential is around minus 70 millivolts. In most textbooks, they're going to have it as minus 70 millivolts. That's not wrong there, because according to what textbook or where your source of information is coming from, they might be slightly different. Some people say minus 65, all kind of thing, right? But minus 70 tends to be, minus 70 millivolts tends to be the more frequent value for the resting membrane potential. All right. So again, remember the sodium-potassium pump is responsible for maintaining this resting membrane potential, which is around roughly minus 70 millivolts. All right. Now there will be there will be some depolarization taking place. All right. Now do you remember what I mean by depolarization? Let's talk about it a little bit. All right. So again, when we were at rest here, so at rest, this will be where the sodium-potassium pump is working. You had a membrane, and on that membrane on the outside, because you are pumping more positive charge on the outside than you're bringing in, the outside was positively charged. All right. And the inside was negative. That is when it's at rest. Now, we say the membrane becomes depolarized. All right, so the membrane is depolarized. So that's here. That represents this region here. Now there's some, we have to be careful. But depolarization is when there's a change in the charge. In other words, the outside goes from positive, now it goes negative, and the inside becomes positive. So we need to see that. So when the inside starts to become positive, in other words, this membrane potential here gets less negative. So you see it starts to rise. Now there's a threshold. All right, there's a threshold for activation. In other words, this membrane potential has to go up to around roughly minus 55 millivolts. All right, so if you look at the graph here, right, this value here should be around minus 55 millivolts. All right, now once you cross that threshold, now if the depolarization, all right, in other words, the membrane potential did not reach minus 55 millivolts, it means that you will not have an action potential being formed. So this is the threshold for activation. The membrane potential must get more positive than minus 55 millivolts. Once it reaches minus 55 millivolts, then sodium ion channels, all right, will open. And when sodium ion channels open, you have an influx of sodium ions coming in. So that is this guy here. So you have an influx of sodium ions coming in. So what's happening is that you're having more depolarization where the inside is coming positive, the outside is negative. So you see the change in potential here is going from minus 70 and it's going up. All right. Now, this will be, so the rise, this rising phase, if you will call it that, the rising phase of the action potential is being caused because of the opening of the sodium ion channel, and sodium ions is coming in. So the inside is becoming positive. All right. So this is the rising phase of the action potential. Now, when it reaches the peak here, what you will see is that this peak will always be of one specific value. All right. So in most textbooks, they'll tell you that this peak here will be around plus 55 millivolts. In some textbooks, they'll have a different number. So plus 55 millivolts. And this peak here is what we call, and it's right here, the equilibrium potential. So this peak here is because of the equilibrium potential. All right. Now, let's see what that is about. Remember, how do these sodium ions come in? They come in based on an electrochemical gradient. So there's two components there. It must have an electrical gradient as well as a chemical gradient. All right. Now look at what's happening here. When the sodium ions are coming in, there's a high concentration of sodium ions on the outside, and there's a low concentration of sodium ions. So there's a huge concentration gradient. So sodium ions will rush in. All right. And in the beginning stages, the outside is positive and the inside is negative. So again, based on just the electrical gradient, the sodium ions will rush in. But as sodium ions rush in, more and more sodium ions come in, here on the inside is now becoming positive, so that's representing here. And when you reach around plus 55 millivolts, what we are saying is that the force created by the chemical gradient to push Na plus in is now being balanced by the electrical gradient that is pushing Na plus out. Alright, so let's recap that. Especially, I can see where you could figure out the Na plus gradient quite easily, the chemical gradient. But look at the electrical gradient. Initially, here was positive, the outside was negative. So Na plus was encouraged, it was forced, the force was good to bring it in. But look at what's happening here. As the inside now becomes positive, this positive charge is going to repel any positive ions from coming in. So you have a force now that is pushing Na plus out. And what we're saying is that they're going to reach a time, which is this maximum peak here, where sodium ions, the force based on the concentration gradient to force Na plus ions in, is exactly being balanced by the force of the electrical gradient pushing Na plus out. So when they reach here, this peak, this is where there's no net movement of sodium ions. So if you look at any other subsequent action potential, you're going to see that the peak is always going to come up here, and we call that the equilibrium potential. Now at this point here is where the sodium ion channels become inactivated. So then there's a loss, or there's a falling phase now of the action potential. Alright, and that is based on the inactivation of the sodium ion channels. Now if you remember from the previous slides, we said that this is where depolarization was taking place. And both sodium ion channels and potassium ion channels would have been activated at the same time based on the depolarization. But the sodium ion channel had a faster kinetic, so they opened faster than the potassium ion channel, so that's why you had the rising phase. Now while the action potential is coming down, the falling phase, alright, this is where potassium conductance will play an important role. Because now, while the sodium ion channels are inactivated, the potassium ion channels are open. So potassium ions are what? How the potassium ions move? Well, if you look at up here, you'll see the answer there, right? So we said that the Na plus was coming in, so that what is that? Is that influx or efflux? You're correct. The Na plus will have come in, so that's influx, alright? And what we're saying here is that because the potassium ion channels, because they have a slower kinetics, right? They activated slower than the sodium ion channels. When the sodium ion channels are closed, the potassium ion channels are open. But look at the direction. The direction is very important. The potassium ions is moving out of the neuron. It's moving from inside to the outside. So potassium ions are moving out. Alright? So what do you think is happening there? It means now that the inside of the neuron, because it's losing potassium ions, it's losing positive charge. The inside of the neuron is getting negative. In fact, it gets even more negative than the minus 70 millivolts. The minus 70 millivolts is at the resting potential. So this area here would represent where it goes even more negative than minus 70 millivolts. We say that is hyperpolarization, and it is the potassium ion channels, the opening of them, that are responsible for the hyperpolarization, where it gets even more negative membrane potential. It goes lower than minus 70 millivolts, and the potassium ion channels is responsible for that. So we call this period here, this area here, the refractory period. All right, so this is the refractory period. So in this area here, there's hyperpolarization taking place. All right, and the last thing I want to talk about in this diagram here is that, let's go back to the equilibrium potential. This could actually, this value here could actually be calculated using this equation here, the Nernst equation. Now the Nernst equation will give you the equilibrium potential for certain ions. And each ion will have a different equilibrium potential because the concentration of that ion outside the cell and inside the cell will be different from another ion. So for instance, like the equilibrium potential for sodium ions will be different from would be a different value from the equilibrium potential from the potassium ions, right? But the Nernst equation, which is this guy down here, could calculate that for you. Alright, so I think I threw a lot of stuff out there to you. Please, if you think you didn't get all of it, again, just rewind, alright, and do it over again. Because I have some slides coming up that we will go through each of these stages once again. But I'll blank out certain words so that just see if you got the true essence of what we talked about here. So please, if you think you missed something again, just go back, try it again. And remember that depolarization is when the membrane potential goes to a less negative value. So if you watch, it was initially around minus 70 and it was going up to less positive value, which is like minus 55 millivolts, which is a threshold. So once you cross minus 55 millivolts, an action potential will be produced. If it is below minus 55 millivolts, an action potential will not be produced. We call this like the all or nothing situation here. All right. So that's depolarization. It's when the negative value here is getting less negative, it's getting more positive. In other words, it's rising until it goes to a peak of around plus 55 millivolts. All right. Now, in the falling phase of the action potential, we have what is called repolarization. So this phase here is what we could call repolarization. And in repolarization is where the membrane potential is becoming more and more negative. All right. So it's becoming more and more negative. Now, this area here, where it becomes even more negative than the resting potential, we call this hyperpolarization. So remember, there's depolarization, there's repolarization, and then there's hyperpolarization. All right, so that's to recap. If you go, oops. So this part here, all of this, is what we could call repolarization. Alright, and obviously the handwriting is much to be desired. Apologize for that. Alright. So again, if you think you didn't get all of it, please give it a pause and go and check it back out again. So if you think you're ready for it, let's go forward. So let's recap. The Na plus and K plus distribution across the membrane. The major intracellular ion is, so it means what is the major ion on the inside compared to the major ion on the outside. So we're looking at, so between Na plus and K plus, which one is the major ion on the inside, or which one is the one with the higher concentration on the inside as opposed to the one on the outside? Alright, and you should have said that the major intracellular ion is potassium ions compared to the sodium ion in the extracellular fluid. And so the natural flow of ions according to their concentration gradient is 4. So you have to choose between Na plus and K plus. Which one do you want to think will leave the cell and which one do you think will enter the cell? So you're correct. Based on their concentration gradients, K plus will want to leave the cell. So it's going from a high concentration, which is inside the cell, to a low concentration of K plus ion, which is outside the cell. And for Na plus ions will want to enter the cell based on its concentration gradient. Now, when an ion wants to leave the cell, we call that efflux. When an ion wants to enter the cell, we call that influx. The movement of positive ions out of the cell leads to the generation of a negative membrane potential, while the opposite is true for positive ion influx. So let's look at the resting potential. And if you remember from the start of this lecture, you know that the sodium-potassium pump is the main guy that is responsible for maintaining this resting potential. And we describe the sodium-potassium pump as being electrogenic. And it's electrogenic because it creates a charge imbalance across the plasma membrane. And it does that by what? So read the rest of that paragraph then and see if you can figure it out. Again, you should have your pencil and paper close at hand. So right, you'll be absolutely correct. It carries 3 Na plus out of the cell for every 2 K plus carried in. Alright, and remember, it's going against the concentration gradient. So for every 3 Na plus ions that goes out, to every 2 K plus that comes in, you'll require 1 ATP molecule because you need energy for this. Alright, and it makes the inside what relative to the outside? That's correct. It makes the inside of the neuron negative relative to the outside. So we say that because there's this imbalance of charge, we say that the membrane is polarized. And what is the average value of the resting membrane potential? What is it normally around? Correct? It's around minus 70 millivolts. And please, it is very important and very critical that you put that sign in. You must put negative. Well, it's a whole different answer. Right, so the action potential. Nerve impulses, also called action potentials, are transient changes in the membrane potential that move rapidly along nerve cells. Action potentials are created when the membrane is locally depolarized by approximately 20 millivolts. Alright, so it goes from a resting value of around minus 70 millivolts to a new value of approximately minus 55 millivolts. When the potential difference rises to minus 55 millivolts, the gates of the sodium ion channels are opened and Na plus begin to flow into the cell. So remember what we said before, minus 55 millivolts is around the, is what we call the threshold, right? So once you go above that, you will generate an action potential. As Na plus enters the cell, the membrane potential continues to increase and additional Na plus channels will be opened so that you get the formation of an action potential. So let's look at what is an equilibrium potential now. A given ionic species continues to flow through a channel only as long as the combination of, so read the sentence there and see if you can figure out what those missing spaces, what those blocks are blocking there, alright? So correct. The sentence should read like this. A given ionic species continues to flow through a channel only as long as the combination of the concentration gradient and electrical potential provides a driving force. So for example, as Na plus sodium ions flows down its concentration gradient, it depolarizes the membrane. Alright? So in other words, the inside is going from negative when it was at rest. So let's remember, at rest, because of the sodium-potassium pump, the inside of the neuron is negative. But as these sodium ion channels open and there is now Na plus coming into the neuron, it will depolarize the membrane. In other words, the inside of the membrane is going to be positive. So it goes from negative to positive. That's depolarization. When the membrane potential reaches approximately, what do you think is that value there? Correct. Approximately plus 55 millivolts. The effect of this membrane potential, in other words, resistance to further entry of Na plus, exactly equals the effect of the concentration sodium ion gradient. Promotion of Na plus to flow inwards. So remember, I was saying before, that there are two things. At the beginning, the electrical chemical gradient is pushing Na plus, and they are both happy to do that in the same direction. Alright? Because at the beginning there, alright, the force that is generated from the concentration gradient And the force that is generated because of the difference in charge is in the same direction. But as more Na plus comes in and the inside becomes more positive, then based on the electrical potential side of things, it doesn't want any more Na plus to come in there, all right? Because positive and positive will repel each other. So the force now is pushing Na plus, based on the electrical gradient, pushing Na plus back out. So what we say that they're going to, this at equilibrium, in other words, when you think about equilibrium, you think about balance. The force based on the concentration gradient and pushing Na plus in exactly equals the force that is pushing Na plus out. So there's no net movement. So we say that that's at equilibrium, and the average approximate value is around plus 55 millivolts. At this equilibrium potential, the driving force tending to move sodium ion is 0. There's no net movement. The equilibrium potential is different for each ionic species because the concentration gradients differ. The membrane potential at which this steady state is achieved can be derived using, correct, the Nernst equation. The falling phase of the action potential then follows as the voltage sensitive channel becomes. So what do you think is blocking there? Correct? So then follows as the voltage sensitive Na plus channels become inactivated. This inactivation is voltage dependent in that it is the response to the depolarizing stimulus but has slower kinetics than the activation process and so occurs later. Alright? So remember, all I is saying there is we're talking about the activation gate and the inactivation gate. Remember, both gates, both activation and inactivation gate, they are both turned on when there's depolarization. That's the stimulus for both of them. Right? The thing is that the inactivation gate and the activation gate have different kinetics. The inactivation gate moves slower. So while the activation gate opens quickly and you have Na plus coming in, after a few milliseconds, the inactivation gate is finally going to reach and block the pore, so no more Na plus could pass through. So it's all about the difference in kinetics, all right? So the inactivation of the sodium ion channels is responsible for the falling phase of the action potential. During this falling phase, a voltage-dependent potassium ion current becomes important as its activation by the depolarization of the membrane has even what? Correct? Slower kinetics than the Na plus channel inactivation. Alright, so what we're saying is that depolarization activates both Na plus ion channels as well as K plus ion channels. But when you look at the diagram that we looked at before, you see that the potassium ion conductance comes a little after the sodium ion conductance. And the reason for that is although they are activated at the same time, and that is at the point of depolarization, the potassium ion channel has a slower kinetics than the sodium ion channel. So the potassium ion channel opens, takes more time to open than the sodium ion channel. So that potassium current comes in a little after the sodium ion current. So this voltage activated K plus channel leads to a brief period of membrane. What is the sodium ion channel responsible for? And that's correct. Hyperpolarization. Alright? So and remember what hyperpolarization means? It means that the membrane becomes even more negative than what it was at rest. So at rest it's around minus 70 millivolts. Because because K plus is leaving from the inside and going out, the membrane on the inside becomes even more negative than normal. So it's what we call hyperpolarization. Alright? And when these sodium ion channels close, the resting potential will be restored by, as you know it, the sodium-potassium pump. So let's just do a little trivia here. Sir Alan Hodgkin and Sir Andrew Huxley originally observed these transient increases and decreases, first in sodium ion permeability and then in potassium ion permeability. For this and related work, Hodgkin and Huxley, along with Sir John C. Eccles, I believe is the name pronounced, won the Nobel Prize in Physiology and Medicine in 1963. So just a little side note there for you. Alright, so we go into the refractory period now. Immediately after the spike of the action potential, there is a refractory period when the neuron is inexcitable. Now, the refractory period could be broken up into two phases. You have the absolute refractory period and you have the relative refractory period. Now, the absolute refractory period means that under no circumstances will an action potential be generated there. So the absolute refractory period occurs at the time of maximal sodium channel inactivations. You're correct. Sodium channel inactivation. While the relative refractory period occurs at a later time when most of the sodium ion channels have returned to their resting state, but the voltage K plus current is well developed. So during the relative refractory period, what we are saying is that an action potential could be generated, but the stimulus would have to be a very strong one. Alright? But during the absolute refractory period, that's when you have sodium ion channel inactivation. You're not going to have any action potential being produced. Now, what is the importance of the refractory period? The refractory period has two important functions. Alright? Action potentials can be conducted only in, correct, one direction. And that direction is away from the site of the first action potential. Alright? They can be generated only up to certain limiting frequencies, meaning that you can't have overfiring of the neuron, in other words. Right, so the all or nothing. The action potential is an all or nothing phenomenon. That is to say, once the threshold stimulus intensity is reached, an action potential will be generated. There are no gradations of amplitude. A given neuron is either at rest or is conducting a nerve impulse. Alright, so remember there was a critical firing threshold. You remember what the value was? Correct, it's around minus 55 millivolts. And we're saying that once you cross minus 55 millivolts, so once the membrane is depolarized from around minus 70 millivolts to minus 55 millivolts, once you reach that value, an action potential will be generated. There's no going back. So that's what we're saying. So there's no, so when you look at a neuron, you're not going to see differences in action potentials, right? They're not going to have a different size or shape or anything like that. They will be the same continuously. So, well, again, I was saying size does not matter, at least for neurons and action potentials, at least, all right? The input of energy all the way along an axon in the form of ion gradients maintained by the sodium-potassium pump ensures that the shape and intensity of the action potential are maintained over long distances. Because nerve impulses display no variation in amplitude, the size of the action potential is not important in processing signals in the nervous system. Instead, it is the number of action potential firings and the frequency of firing that carries specific information. Right? So it is the frequency, not the size, but the frequency of the action potential. So let's see. Action potentials resulting from a strong and a weak stimulus differ in their frequency. A strong stimulus produces a rapid succession of action potentials, each one following along the axon just behind its predecessor. A weak stimulus results in fewer action potentials per second. So in other words, a strong stimulus will have a high frequency, of action potentials, while a weak stimulus will have a low frequency of action potentials. Moreover, right, a strong stimulus is likely to stimulate more neurons than a weak stimulus. While a weak stimulus might result in action potentials passing along just one or two neurons, a strong stimulus could produce an action potentials in many more neurons. The brain can therefore interpret the frequency of action potentials arriving along the axon of a sensory neuron and the number of neurons carrying potentials to get information about the strength of the stimulus being detected by that receptor. Alright, so it's all about the number of neurons being activated as well as the frequency of these action potentials. Right? Another thing that is important is the positions, right, which neurons are being stimulated. The nature of the stimulus, whether it is light, heat, touch, or so on, is deduced from the position of the sensory neuron bringing the information. If the neuron is from the retina of the eye, then the brain will interpret the information as meaning light. Alright, so I'll give you an example below there. Now, please do not go rubbing your eye and trying this. You just take my word for it. If for some reason a different stimulus, such as pressure, stimulates a receptor cell in the retina, which is a part of the eye that is responsible for interpreting light, the brain will still interpret the action potentials from this receptor as meaning light. This is why rubbing your eyes when they are shut can cause you to see patterns of light. So again, take my word for it. Right, so I'm hoping that I still have most of you all here with me. Alright, I know it's a lot. And again, you all devise the ways in which you want to study this thing. If you want to take it like 10 minutes and then stop, relax, come back again, that's up to you. There's a pause button in YouTube there that you could use as much as you want. Alright, our next thing to do is to keep a pen and paper and just keep writing down these notes. So the more you write, the better you will remember. Right, so the next thing now is the propagation of an action potential. Alright, so far we have spent a lot of time looking at the generation of an action potential. Now we're going to be looking at the propagation of an action potential. Now in this diagram here, they have an action potential going up to plus 30 millivolts. So we'll use that for this diagram here. The resting potential is the normal value, which is minus 70 millivolts. So in other words, in A here, alright, so let's see if we can just single out that for now. So in this area here, an action potential is being developed. So right now, at B and C, they are at rest. So they have the resting potential. So you see the outside of the membrane is positive. The inside of the membrane is negative. This here is the bilayer. Alright. So this at C is also at rest. It's positive outside the membrane, negative on the inside the membrane. Now if you watch here, an action potential is being generated. The outside is, so this part of the membrane here is depolarized. The outside is negative, the inside is positive. So all here is a region of depolarization. And when there's depolarization, sodium ion channels will open. So you see that Na plus is coming in there. Alright, so an action potential is being developed. Now what will happen is that you have differences in charges. You have a region here that is negative, while you have on the side of it, you have a region here that is positive. And likewise, on the inside, you have a region here that is positive, but neighboring to it has a negative region. So what we say is happening here will be that local currents, local circuits will be set up. Alright, so local circuits will be set up here. Now in region B now, look what's going to happen. Positive ions, positive sodium ions, will move over to this side. So it's going to be attracted to this negative charge here, so the positive sodium ions are going to move over here. And positive sodium ions that are in the A region is going to move over to the B region. So what you're going to think is going to happen there? When these positive ions move from the A region to the B region, it means that here starts to get more positive. In other words, here on the inside of the membrane at B starts to get less negative. So instead of it being minus 70 millivolts, which it originally was, because the inside here is getting less negative because of these sodium ions moving over to this area, it goes to minus 55 millivolts. And what's special about minus 55 millivolts? That's correct, that's the threshold value. So once these sodium ions, they move over to this region here and becomes minus 55 millivolts, sodium ion channel is going to open in this region and sodium ions will flow in, and an action potential will occur. And then when this happens here, then a local circuit is going to be set up here, and again you're going to have movement of ions, and an action potential will eventually be produced at C, and you keep going like that. Alright? Now the thing is that the direction in which the action potential will be going, it will be in this direction here, right? So the action potential will always be going in this direction. The reason for that is that here is where the local circuit will be set up. In region A, a local circuit will not be set up. In other words, the action potential will only go from A to B to C. It cannot go back to A. And the reason for that is that A would have been going through its refractory period. Alright? So because A was going through its refractory period here, there's no way that an action potential would have been initiated here. So it has to go in this direction. Alright? So you see, let's read here. The depolarization due to the action potential B spreads electronically by local current flow through the axon, depolarizing adjacent areas of the membrane toward threshold, which is around minus 55 millivolts, triggering an action potential at C. Alright, so that would be C, will be this fella here. Alright, well, in this case, it will be this one here. Alright, voltage gated Na plus channels at A, which is here, is inactivated. Alright, so why? Because it's going through its refractory period. Alright, so a new action potential is not initiated A. So there's no action potential A because it's going through its refractory period. So it keeps going forward. The action potential is propagated forward. Alright. Now here's the next thing. We are looking at these local circuits being set up next to each other. Now if this was covered with a myelin sheet, it means that here it will be insulated, there won't be no charge. So you have to go to the end of the myelin sheet where they have the nose around here for you to get a local circuit being set up. So that's why we say you could speed up the action potential by because of myelin sheets being present. So let's see if you remember that. These changes in potential in one part of the axon are rapidly passed along the axonal membrane. The Na plus that rush into the cell in one localized region actually diffuse further along the axon, raising the concentration of sodium ions and depolarizing the membrane, causing sodium ion gates to open in that adjacent region of the axon. In this way, the action potential moves down the axon in a wave-like fashion. This simple process has several very dramatic properties. Action potentials propagate very rapidly, alright, up to and sometimes exceeding 100 meters per second. So that's amazing. The action potential is not attenuated as a function of distance transmitted. In other words, the action potential doesn't get smaller as the distance increases. Alright? So look at this nice cute guy. Or cute, well, I guess this is according to the eye of the beholder. But this is a puffer fish. Alright? And puffer fish is big in Japan. It's a delicacy in Japan. And the dish that is served, they call it fugu. Now here's the thing, puffer fish is very poisonous. Right? And the toxin that they contain is concentrated in the skin and sexual organs. Alright, and this toxin has to do with something, with all that we have been doing before. Alright, so this toxin, it affects the production of an action potential. Alright, now, so it's concentrated in the skin and sexual organs of the pufferfish. So there's a little background information here. Like in World War II, when after the war, you know, there was a period of poverty and so on. So people in Japan would have gone by the dustbins, by restaurants and stuff, and try to get any kind of scraps and eat them. And unfortunately, they might have had some remains of the pufferfish. And when they grill that and they eat that, they died. All right. So I think there was a lot of deaths but with the misuse of pufferfish, right? Because of the ingesting the toxin. In fact, it has been made law now that any chef cooking pufferfish, they have to get a certain license indicating that they have been trained in preparing the fish. All right, so it's very, but luckily now I think the death rate on pufferfish last time I read was that there's like 3 deaths per year in Japan, and mostly is because of people preparing it on their own. All right. Now, it's a delicacy, as I said. I think you could get a dish for like 100 US or more in Japan. People say it's the best sushi out there because of the tenderness of the meat. But another thing about the meat is that it has the toxin. It has a lower quantity of toxin in the meat itself. So you get that lightheadedness and you get this nice feeling and all of that. All right. There's no antitoxin or no current treatment for this for the toxin that's in the pufferfish. All right. In fact, back in the day, well, what happens is that when you ingest the toxin that the pufferfish contain is the tetrodotoxin, and basically what that toxin would do is that when you ingest that, it shuts down the body, your respiratory system, your heart, everything stops. All right, and it brings you to that point that well, according to the concentration, it could either bring you to that point that is close to death, or even, God forbid, you'll die. All right, so it has been shown that some people, according to the dose that they took, they were still alive, and but you look physically like you are dead, but you are still alive. And back in before all of the medical advancements and stuff, like if they suspected that you had that you ingested pufferfish and that's why you're in this state, they wouldn't even carry out an autopsy or anything, or they wouldn't bury you. They'll just keep you there and wait a few days to see if you wouldn't get up. Because once the toxin is metabolized in the body, you will get up, you know, providing the concentration, right? In Haiti, you could do some research about it. The witch doctors and so on, they use that toxin as well in specific amounts to do what is called zombification, and to also you know, to show people that they have like supernatural powers. So I guess if they want, they would give somebody the toxin itself, and then they would show that they could bring them back alive after a few days. So you can read up some interesting stuff about that. So let's talk about the toxin itself, tetrodotoxin, or what we could just simply abbreviate it as TTX. The Na plus channel is so essential to successful body function that it has become the target in evolution of several potent poisons. The pharmacology of these agents has provided important insights into the further definition of functional regions of the channel. So in other words, we have used this toxin, this tetrodotoxin that is found in the pufferfish, to really look at our and study the ion channel. Now, at the outer end of the sodium ion channel pore is a site where the pufferfish poison tetrodotoxin, TTX, a small lipid insoluble charged molecule, binds and blocks sodium ion permeability. Now, let's look at what's happening here. You see this that's in red? That's the most important part of the tetrodotoxin molecule here. All right. So tetrodotoxin has a cationic, and what we call this group here. Now it's cationic because of this positive charge, right? Positive cation, right? So what we call this group here in biochemistry, people, when you look at this. All right. So I hope you said guanidino. All right. So tetrodotoxin has this cationic guanidino group and is effective only when applied to the external surface of a neuron. Their injection into the cytosol elicits no response. It is therefore thought that these toxins specifically interact with that's correct, an ionic carboxylic group, so ionic meaning negatively charged, located at the mouth of the sodium ion channels on its extracellular side. Pairs of amino acid residues required for high affinity tetrodotoxin binding are located in analogous positions in all four domains near the carboxyl ends of the short membrane reentrant segments between transmembrane alpha helices from helix 5 and helix 6, the P loops. All right. So other sodium ion channel blockers, we have local anesthetics, lidocaine and procaine. You have lipid-soluble steroids found like in the golden poison frog. All right, so that's those guys there in the background. So frog skin poison, BTX. You have the plant alkaloids. All right, and the last one there from marine dinoflagellates, you have saxitoxin. All right, so it's pronounced saxitoxin. I remember a colleague talking about it as sexy toxin. No, that's not right. It's saxitoxin. All right, so if you look at this diagram here, you see where different toxins, where they bind and when they inhibit. I will just again show you TTX. So you see that it binds there at the mouth there, the selectivity filter, sorry. All right, binds to some negatively charged carboxylate groups, and therefore no Na plus could flow through. All right, so it inhibits that. So let me put forward a question for you. Why in the course of evolution have ion channels become the preferred target of toxins rather than some critical metabolic targets such as an enzyme essential in energy metabolism? So that's a pretty good question for you to answer. So let's go ahead. Ion channels are extraordinary amplifiers. Opening of a single channel can allow the flow of 10 million ions per second. Consequently, relatively few molecules of an ion channel protein are needed per neuron for signaling functions. This means that a relatively small number of toxin molecules with high affinity for ion channels acting from outside the cell can have a very pronounced effect on neurosignaling throughout the body. A comparable effect by way of a metabolic enzyme typically present in cells at much higher concentrations than ion channels will require far more copies of the toxin molecule. Alright? So again, why is it that toxins will rather target ion channels as opposed to enzymes? Well, there are two things. Alright, just to remember. One is that there are a lot less ion channels than enzymes. It means that you need a lower concentration of the toxin if you're blocking ion channels as opposed to enzymes. And then the next big thing is that these ion channels are on the outside of the cell, right? In other words, the toxin doesn't have to design mechanisms or the molecule itself has to be nonpolar for it to go through into the cell. All right? Enzymes are within the cell. So for this toxin to get to the enzyme, you would have to develop ways to enter the cell. While with ion channels, the toxin doesn't have to worry about that. All right? It doesn't have to get into the cell to get to the ion channels. So what is your homework now? You need to tell me what are glial cells, what are Purkinje cells, and tell me what is the mechanism of action for BTX and saxitoxin. Alright, so check out that homework. And a thought for the day. So think of this carefully and be careful. Alright guys, so this was a long one. I apologize for that, but I think the information here was very important that you know about it. Again, you have the pause button there. So please, if it's too much in one go, that's alright. Break it up in parts. It's your thing, so you do whatever it will take to understand this topic. Alright? And if you like what you heard, alright, show your love, hit the like button on YouTube, as well as subscribe to the channel so you get all the new videos that's coming up. And please, if this helped you in your exam and so on, let me know. I'd love to get some feedbacks to encourage us to go produce more videos. Right? If you need to ask me a question, if you get any problems understanding anything, please send me an email at this address, and we could talk more about it. If you have some interesting stories or you have some interesting information on your side, please send it to us. We'll share. And as usual, guys, these videos were designed for you all to better your studies and so on. So thank you for listening. It's really a pleasure to design these videos for you all. So good luck in your studies.